We're going to be talking about DNA in the last bit of this semester, specifically looking at sections 8.1, 8.2, and 8.3 leading up to our final exams. So to start with, we're going to be reading about identifying DNA as the genetic material. Going back in history, we didn't know that it was DNA that coded for our genes, which make up our physical traits, hair color, eye color, those sorts of things. We knew that DNA was there, we just didn't entirely know what it did. People initially thought that it was proteins that dictated our physical appearances, our traits, not necessarily the DNA. So our key concept for 8.1 is DNA was identified as a genetic material through a series of experiments. Some of the names that you're going to be seeing as we go through this include Griffith, Avery, Hershey and Chase, and there's going to be a few others as well. Okay, if you care to read this out of the book, it can be found on page 226. Alright, some people think a complicated answer is better than a simple one. If they have a head cold, for instance, they may use all sorts of pills, syrups, and sprays, when they simply need rest and water and warm chicken soup. In the early 1900s, most scientists thought DNA structure was too repetitive for it to be the genetic material. Proteins, which are more variable in structure, appeared to be a better candidate. Starting in the 1920s, experiments provided data that did not support this idea. By the 1950s, sufficient evidence showed that DNA, the same molecule that codes for GFP in the glowing mouse, carries genetic information. Okay, DNA, it only contains 4 nucleotides, which you may be familiar with if you've heard A, T, C, and G. Those are nucleotides in DNA. That's it. That's it. We'll be looking at the actual structure of DNA in 8.2, but because that's all there was to DNA, people thought, oh, it's too simple to create the variety of traits that humans and all living things have. So it's got to be proteins. They're more variable in structure. They seem to be more complex, so they're probably a better candidate. So we're going to look at how we discovered that it was actually DNA and not proteins. The first person we're going to be talking about and the main idea is Griffith, and he found a transforming principle. So think about what transformed means. In 1928, the British microbiologist Frederick Griffith was investigating 2 forms of the bacterium that causes pneumonia. One form is surrounded by a coating made of sugar molecules. Griffith called these bacteria the S form because colonies of them look smooth. The second form of bacteria do not have a smooth coating and are called the R, or rough form. As you can see in figure 8.1, when Griffith injected the 2 types of bacteria in the mice, only the S type killed the mice. When the S bacteria were killed with heat, the mice were unaffected. Therefore, only live S bacteria would cause the mice to die. So let's take a look at what he did here. Here's our 2 forms. We have our S form and we have our R form. Remember, this was apparently a smooth form, although this picture doesn't do a great job showing it. And this bacteria here was speckled a bit more, so it was a little bit more rough. And both of these are 2 types of bacteria. Both cause pneumonia, but this strain of pneumonia, a different form of pneumonia, would kill a mouse, whereas this one would be pneumonia that a mouse could live with and recover from. So what he did is he took this strain, injected it into mice, the mice died. He took this strain, injected it into mice, and the mice lived. Then Griffith took this strain right here that would kill a mouse, and he heated it up. So think about like when you cook your meat. You cook your meat to a certain temperature to make sure all the bacteria is dead inside and that you can safely consume that meat. Well, he did that with these bacteria. He heated them, he killed them, and then he took this S bacteria, which previously killed a mouse, injected it into mice, and the mouse lived. Why? Well, because this bacteria was now dead. But something amazing happened. and this is what Griffith called the transforming principle. He took this S bacteria that was now dead, and he put it in a jar with this bacteria and mixed the two together. Keep in mind that separately, the R bacteria, the mouse lives, the S bacteria that has been killed, the mouse lives. But when you mix them together, the mouse dies. So what he's thinking is something in this bacteria, although alone it let a mouse live, something was passed off from this guy to this guy, which now is alive, causing this mouse to die. And that's what we see going on here. Okay, Griffith next injected mice with a combination of heat-killed S bacteria and live R bacteria. To his surprise, the mouse died. Even more surprising, he found live S bacteria in blood samples from the dead mice. Griffith concluded that some material must have been transferred from the heat-killed S bacteria to the live R bacteria. Whatever that material was, it contained information that changed harmless R bacteria into disease-causing S bacteria. Griffith called this mystery material the transforming principle. Why? Because this red bacteria was transformed into a lethal form because something from this dead bacteria was passed on to it. So this red one was transformed, becoming lethal. Next section, main idea. Avery identifies DNA as a transforming principle. Keep in mind that it was Griffith that discovered the transforming principle, but he didn't know that it was DNA. Avery discovered that it was DNA. What exactly is the transforming principle that Griffith discovered? That question puzzled Oswald Avery and his fellow biologists. They worked for more than 10 years to find the answer. Avery's team began by combining living R bacteria with an extract made from S bacteria. This procedure allowed them to directly observe the transformation of R bacteria into S bacteria in a petri dish. Avery's group next developed a process to purify their extract. They then performed a series of tests to find out the transforming principle was DNA or protein. Qualitative tests. Standard chemical tests showed that no protein was present. In contrast, tests revealed that DNA was present. Chemical analysis. As you can see in figure 8.2, the proportion of elements in the extract closely matched those in DNA. Proteins contain almost no phosphorus. DNA does contain phosphorus, so if phosphorus is present, it's coming from DNA because proteins really don't have any phosphorus at all. So something containing phosphorus was passed from the S bacteria to the R bacteria. Couldn't have been protein. Enzyme tests. When the team added to the extract enzymes known to break down proteins, the extract still transformed the R bacteria into the S form. Also, transformation occurred when researchers added an enzyme that breaks down RNA, which is another nucleic acid. Transformation failed to occur only when an enzyme was added to destroy DNA. So enzymes are things that break down stuff, and there's different types of enzymes. There are enzymes that will break down proteins and only proteins, and there are enzymes that will break down nucleic acids, DNA and RNA, and they'll break down only nucleic acids. So when researchers added enzymes that broke down proteins, then the protein could not have been passed off from the S form to the R form, and the bacteria was still able to undergo transformation. However, when we broke apart the nucleic acids and we mixed the two types of bacteria together, nothing happened. So this indicated that it had to have been DNA and not proteins, because when we broke down proteins, making them non-functional, transformation still occurred. In 1944, Avery and his group presented this and other evidence to support their conclusion that DNA must be the transforming principle or genetic material. The results created great interest. However, some scientists questioned whether the genetic material in bacteria was the same as that in other organisms. Despite Avery's evidence, some scientists insisted that his extract must have contained protein. Well, You can insist that it contains protein, but if it's all broken down, how does it contain protein? And our last main idea, Hershey and Chase confirmed that DNA is the genetic material. So we have Griffith discovering that there's a transforming principle, Avery going through experiments proving that it's DNA, and Hershey and Chase saying, yeah, Avery was right. So we have scientists working off of other scientists' findings. Conclusive evidence for DNA as a genetic material came in 1952 from two American biologists, Alfred Hershey and Martha Chase. Hershey and Chase were studying viruses that infect bacteria. This type of virus, called a bacteriophage, or phage for short, takes over a bacterium's genetic machinery and directs it to make more viruses. Phages, like the ones Hershey and Chase studied, are relatively simple. Little more than a DNA molecule surrounded by a protein coat. This two-part structure of phages offered a perfect opportunity to answer the question, is the genetic material made of DNA or protein? By discovering which part of a phage, DNA or protein, actually entered a bacterium, as shown in figure 8.3, they could answer this question once and for all. Hershey and Chase thought up a clever procedure that made use of the chemical elements found in protein and DNA. Protein contains sulfur, but very little phosphorus, while DNA contains phosphorus, but no sulfur. The researchers grew phages in cultures that contained radioactive isotopes of sulfur or radioactive isotopes of phosphorus. Hershey and Chase then used these radioactively tagged phages in two experiments. Experiment 1. In the first experiment, bacteria were infected with phages that had radioactive sulfur atoms in their protein molecules. Hershey and Chase then used an ordinary kitchen blender to separate the bacteria from the parts of the phages that remained outside the bacteria. When they examined the bacteria, they found no significant radioactivity. Experiment 2. Next, Hershey and Chase repeated the procedure with phages that had DNA tagged with radioactive phosphorus. This time, radioactivity was clearly present inside the bacteria. From the results, Hershey and Chase concluded that the phage's DNA had entered the bacteria, but the protein had not. Their findings finally convinced scientists that the genetic material is DNA and not protein. So looking at this picture, let's talk about what they did. This is a bacteriophage, and this is a bacteria. We already know that bacteria are really small, so if this is a bacterium, then this bacteriophage is really small. What we see on this image here, the red part, that is the outer part of this bacteriophage. This outer part is made up of protein. Protein contains sulfur. What we cannot see so well is the DNA that's inside this bacteriophage. The DNA inside the bacteriophage that we cannot see does not contain sulfur, it contains phosphorus. So what they did, first off, they took the protein outer part right here on this bacteriophage and they made it radioactive. So think of radioactivity, something that glows. They let this bacteriophage infect this bacteria. Well, if this bacteriophage is passing on protein to the bacterium and this protein is glowing, then it should glow inside the bacterium too. But they didn't find that. There was nothing glowing inside this bacterium after the bacteriophage had infected it. So that ruled out protein right there. If it's glowing and the glowing part is passed on, then we should see glowing. So they tried taking the DNA. The DNA contains phosphorus, and they made DNA glow. They made it radioactive. What they found is after making the DNA inside this bacteriophage glow, there was glowing going on inside the bacteria, meaning that DNA was passed from the bacteriophage to the bacterium. This confirmed that DNA is the genetic material, and it is definitely not the protein.